We are recording now. So let's go ahead and pop in here. And slideshow.
Is the power?
Load the PowerPoint into Canvas, but for whatever reason, it's, I guess, like full of storage space. So I sent it as an email attachment. So check your inbox. And if it's not there, let me know and I can resend it out, but you should see that in there. You all found it in your inboxes? Yeah, cool. So yeah, go ahead and take a second, try and find that. Morning.
Thank you.
Yeah, great, thank you. All right, y'all, so I'm already getting spotty internet service, so bear with me. We're gonna stay patient like always. So the exam, we're gonna talk about this for the first, 15, 20 minutes of class. We're gonna go over some of the questions. I'm gonna show you the questions that you couldn't see. And we're gonna talk about what we're gonna do moving forward for the second half of this course before we even get into today's material. So the first intro part is gonna be like 20 minutes, maybe 15, 20, we'll see how it goes. So on average, people did not do as well as I expected. The average score was a 58. The highest score was a 92, so somebody did get an A, but you can see here with the spread that most of the people did like C's or D's or less, right? So that's an issue, that's a problem. So we need to move forward with that. One thing to notice is that the average time was 38 minutes. You had 100 minutes to complete this. Maybe you just don't know the answer, so you just submit your exams, but you have more than double the amount of time available, double, triple, quadruple check your answers. Okay, so I expected people to cheat, and they did. This is an online course for your exams, so I know that somebody's going to use a book. So let me tell you what I did so that you know that I know. So first off, let me say, 4 people cheated. They're all going to get away with it because I don't have direct video evidence of you with a book in front of the camera. I have video footage of you flipping through the book. It's just not recorded on camera. So you got away with it. You've disrespected me and your classmates, but most importantly, you've disrespected yourselves. So keep that in mind. Please don't do that anymore. You're going to end up killing somebody in a hospital if you cheat. That's my mentality. Okay, so another thing. Saw 3 people doing this. You're reading every question and answer, and then looking in the corner. You're clearly on FaceTime, okay? Please don't do that. So now that I know that you're doing it, you know that I know, don't do it anymore. Also, if I do catch you doing it, you will receive a 0. So that goes for quizzes and exams. No tolerance. So papers, these are going to do by Thursday. Let's try and get it in the morning. If you need more time, shoot me an email. I'm going to try and get those graded on Thursday, Friday, over the weekend as soon as possible. I start teaching at Bellarmine this week, so I'm going to be very busy. So I'm going to try and get as much done this week as I can so you get your grades back. Okay, so here's what I'm thinking about doing moving forward. Since we often have an extra 20, 30 minutes at the end of class, what I'm thinking about doing is spending the first 10, 15 minutes reviewing the prior day's content. So tomorrow we'll come in and we'll get a 5 to 15 minute review over what we talk about today and keep doing that on a daily basis. So that's an option. Or we could do Thursday reviews on Thursday morning for maybe 30 minutes and then take your quiz. Well, we don't really have time for that. So this is probably the only way that we can do some sort of a daily review. How does this sound? Yeah? Cool. Would you rather have it at the beginning or the end of class? Beginning? Okay. So that's what we'll do. So that would be a good time for you to come up with any questions. So that way you come in and you say, hey, talk about endotoxin again. And I'll be like, I got you. Let's talk about it. So that's what we're going to do just to keep everybody on the same page, because I know this is a really fast-moving class. This is how I think we can stay together a little bit better. Okay, so that took all of 6 minutes. That's not bad. All right, so let's talk about today's content. Today is a lot of definitions more than anything. Today's a very big picture kind of day. Let's just take a look at our table of contents and see where we're at. Okay, so tomorrow we're going to start talking about the immune system. Okay, so the rest of this week we're going to talk about the immune system. So today we're talking about general infection, and then we're going to get into how our bodies respond to that throughout the week. It's going to be a lot of fun, and then we'll get into diseases. All right, so feel free to slow me down, stop me at any point. So we are born into a world full of microbes. We're going to get them as soon as we're born. The word infection is going to refer to the entry and multiplication of one organism inside of us. We're going to call ourselves the host. And then we respond to the infection through a series of inflammatory reactions. And this is generally going to be at the site of the infected tissue, but keep in mind that certain infected tissues can be systemic, like the blood. So you can have systemic inflammation as well as localized inflammation. All right, let's take a look at this figure real quick. So this shows infectious diseases that cause death worldwide. You'll notice that cardiovascular disease is not on here. Cancer is not on here. Give it a couple of decades, and we're going to see that more of these long-term chronic diseases are caused by pathogens. Cardiovascular disease is the leading cause of death globally, and we do know that certain bacteria and viruses can at least play a role in cardiovascular disease. So again, give it a couple decades and I expect this pie chart to change. And that's the beauty of science, is that it is tentative. We are always willing to accept new information. So in dealing with the infection, so we have an inflammatory response, and if we are unable to deal with that infection, we can say that we have disease. Look at the word disease. We brought this up a couple times, but your book finally describes it. So the microbiome is the collection, so the complete population of microbes in the body, and we can split this up into different body regions as well. You'll have heard of the gut microbiome, probably heard of the skin microbiome. So these are all subcompartments of the whole microbiome. So we are covered inside and out, head to toe, in bacteria, and we need them. If we didn't have them, we'd probably be in some sort of a state of disease, right? So we call this symbiosis. We live together with our resident microbiota. We are together living, therefore we are symbiotic. And so then we can break this symbiosis down into a couple different terms. Let's say that we have certain skin organisms that help us combat infections, and maybe they receive some sort of a benefit from our sweating or from our body temperature. We would call this a mutualistic relationship, where both the host and the microbe benefit. So a win-win situation. So before we go further, something else I want to keep in mind, or for you all to keep in mind, is that within the microbiome, one organism can be mutualistic for another bacterium. So we have mutualisms between host and microbe, but also between microbe and microbe. And so let's say that one microbe can be within or on us, and it benefits, but doesn't hurt us or give us any benefit. We could say that we are in a commensal relationship. We describe the organism as a commensal organism. And so keeping in mind that our gut is full of microbes, both good and sometimes bad depending on the situation. Things like antibiotics can destroy our resident gut microbiota. Also, diet. I'm surprised the book doesn't mention this. If you eat a poor diet, you can destroy your healthy population. Alternatively, you can restore your gut microbiome by eating a healthy diet as well. So eating foods high in fiber, high in nutrients, you can restore your gut microbiome. And so the gut microbiome, I was reading in your book, they say that some E. coli strains can produce vitamins B and K. Health. Sharing back on. Go down. Okay. All right. We'll start with this figure. So we are born either through vaginal birth or through a cesarean section, otherwise known as a C-section. So if you're born through a vaginal birth, then you're going to pass through the maternal vaginal canal, and you're going to receive those gut microbiota, or rather those vaginal microbiota along the way. I assume that the baby gets some in the mouth. It'll colonize the oral cavity. It's also going to get on the skin. It can get in the eyes, which is a good thing, right? This is protective. You can get species such as lactobacilli, Bacteroidetes, bifidobacteria. So that's after birth, or pardon me, that is during birth. After birth, things like nursing. So consuming your mother's milk gives you a nutrient source, but it also gives you immune protection. Alternatively, if you're born by a C-section, then what I am pretty sure doctors do is they will take a swab of the vaginal microbiota and probably put it in the mouth of a baby, possibly on the skin, maybe in the eyes, I'm not sure, to inoculate the baby with a healthy microbiome. Again, I don't want us to think about bacteria as dangerous at all. We need bacteria. And then baby's fed formula, so they're probably not going to get the essential nutrients. Maybe, I don't want to say that. They're not going to get the natural nutrients that a mother would provide. Maybe certain sugars found in milk, or definitely certain immunoglobulins, so immune proteins, would not be found in formula. All things to consider as young adults. All right, so a pathogen. Pathogen is a virus, some sort of a Let's see, a virus, a bacterium, a multicellular organism, a fungus that is going to get in us and cause harm. A parasite is going to cause damage. So a parasite is a pathogen. We can describe the ability of an organism to cause disease, talking about its pathogenicity. So gen referring to generate, to create, pathos being an illness, something bad. So can you create a state of disease? And so we'll refer to pathogenicity by talking about virulence. So virulence means the ability to cause damage. Anything bacteria, viruses.
It says in the book
sorry,
that Candida, I'm sorry. It says in the book that Candida albicans is labeled as moderately virulent.
Okay.
Is that how you say that? Virulent?
Yeah, virulent. So what would be an example of a virulence factor? What allows Candida albicans to be virulent or to cause disease?
Is transmission rate?
Okay, sure. So if something can divide rapidly or transmit from one host to another, that is a virulence factor. Great job. So things like spike proteins on a coronavirus. That's going to allow for attachment to a cell. You need to attach before you can enter. So anything that has to do with infection is going to be a virulence factor. All right, so within the genome of microbes, these genes that encode for virulence factors are often found next to each other, and we would call that a pathogenicity island. All right, we have another slide coming up with more details about this figure. Let's start with some more definitions here, though. So an exogenous infection is going to be when a pathogen is going to get inside us. So COVID-19, this is an exogenous infection. We don't have SARS-CoV-2 in us waiting to cause disease. We get it from the environment. So exo, meaning outside. Same thing would be influenza, rhinovirus that causes common cold. All of these are exogenous. An endogenous infection is going to occur when our normal microbiota get into what should be a sterile site. So let's say that you are, I don't know, just walking and you catch your arm on a rusty nail. So you could get some tetanus from the rust, but let's say it wasn't rusty. Some of your skin bacteria can get in your blood. They don't cause disease when they're on the skin, but when they get in the blood where they shouldn't be, they can. So that would be an endogenous infection. And then opportunistic infections, this is really applicable to you all as pre-healthcare workers. So this is going to be when our normal microbiome senses the change in the environment and causes disease. So can anybody think of opportunistic pathogens? or a disease caused by an opportunistic pathogen. Think like pneumonia. So I believe this is happening in COVID patients. So they get COVID-19, and then they get what's called a secondary infection where their normal lung microbiota promote a pneumonic state, or pneumatic. They get pneumonia because their immune systems are dealing with another infection. So people who are immunocompromised are prone to opportunistic infections. I want you all to know that. So what can cause immunocompromisation? We'll think of maybe 2 or 3 things. What can cause a lower immune system in a patient?
Diabetes, health disease.
There you go. So diabetes, I'm not quite so sure about. A more clear-cut example would be something like human immunodeficiency virus, or AIDS. Let's try and think of another example specific to women. Pregnancy? Yeah. Women who are pregnant have all right. Let's go here. All right, so primary versus secondary infections. I'll just reiterate. Primary infections take place at an otherwise healthy spot. So you breathe in the influenza virus, and now your lungs are infected with the influenza virus. They were healthy, now they're not. Secondary infection, you have HIV, and now your healthy lung microbiota cause you to have pneumonia. Primary versus secondary. Diseases can be local or systemic. So with the pneumonia example, the local is the lungs. Or with the skin abrasion, it's at the skin. But let's say you get that tetanus infection and it goes in your blood, now you have a systemic infection. So it's going to go all around the body. Metastatic cancer is also an example of a potentially systemic disease. So as it travels from one location to another, cancer can spread through the body. All right, let's talk about these phases of an infection. Let's get our pointer back. All right, so at the very beginning, we are going to acquire an infection, and we're going to do what's called incubate, or rather the microbe will incubate inside of us. And so this is going to be the time before you start to see symptoms. So you acquire an infection, you might not see anything happen for a day or 2. Then during the prodromal phase, this is where you might start to see something like, I don't know, a runny nose, a fever, some sort of a symptom. Responses to infectious agents are not always bad. You don't always want to treat symptoms. When you have something like a fever, I wouldn't treat it unless it gets to be like a really, really high temperature, because that fever is killing off things that cause disease in your body. So symptoms can be protective. They can also be detrimental. All right, during this acute phase, this is going to be when the illness is most severe. So this is going to be when your symptoms are really, really present. going into the decline phase here. So this is going to be probably after 2 weeks. It takes about 2 weeks for our B cells to make what's called a memory cell that is going to produce antibodies. So antibody factories take about 2 weeks to get rolling in our bodies. And then after we have dealt with the infection, we'll have this return to good health called a convalescence phase. So again, with antibodies taking about 2 weeks to produce, that's why whenever you all travel, we also, we often have a 2-week incubation period or isolation period where you're supposed to not be around other people so that you can deal with your potential coronavirus infection. All right, so we talked about symptoms. A symptom is going to be presented by the host. So a fever, a runny nose, achiness, malaise, all these sorts of things. A sign is something that is measurable. So I can take a blood sample and look for and count bacteria in my blood. Or take a throat swab and count the number of different microbial species present. These are signs. And then we can put the signs and the symptoms together and call that a syndrome. So that would be, all right, you are presenting to me, you've not felt good for 3 days, you have a sore throat, I got a positive strep test, and you are sleepy all the time. Therefore, I'm going to say you have this syndrome, this collection of signs and symptoms. And so we can characterize syndromes so that we can have adequate treatment plans for whatever the illness is. And so if we're treating diseases that are referred to as acute are going to be short-lasting. These are much easier to treat. Again, something like influenza, bronchitis, right? These are all diseases that you can get and then we can treat. Chlamydia is a sexually transmitted disease that can be treated readily, whereas syphilis is more of a chronic disease that can last for decades. So chronic diseases, this says linger for long periods. I want you to think that these can take decades to develop. Something like cancer, something like heart disease. These can start in your teenage years but not manifest until your 40s or 50s. And so it's hard to treat something if it takes decades to cause a disease. So examples of chronic diseases, like I said, heart disease, type 2 diabetes, cancer, more difficult, but are probably, at least in some fashion, caused by microbes. All right, so we have 5 words to describe some blood infections. So if we have 5 words on a single slide that have to do with one thing, this is a great candidate for a multiple choice question. Bacteremia is where you get what we call a transient blood infection. So that means you floss your teeth and some bacteria get in your blood, but then they're gone in like 30 minutes because your immune system handles it. So yeah, you had a little blood infection, but it was quick, it was over and done with. We call that bacteremia. So if you see emia, think blood. Septicemia is a full-on blood infection. So instead of a transient 30-minute in and out kind of infection, this is where no, your gums or your intestine has some sort of a tear in it, and you're always having bacteria in your blood. Septicemia. Our response to that is called sepsis. This is a systemic inflammatory response to septicemia. You've probably heard of people being in septic shock. So that's where you just have an enormous blood infection and your body is trying to fight it, but it's everywhere, so you go into some sort of a shock. And then just based on the words, you should be able to see that fungemia and viremia are fungal and viral blood infections, respectively. So what I want you to know from this is probably the difference between bacteremia and septicemia. That's what's most important here. So which of the following, let's take sepsis out, give you the top two, bottom two. Which of the following is a transient state of a blood infection? Bacteremia. So that's probably going to show up on quiz 3 or exam 2. All right. This is figure 14.5 we were at about 10 minutes ago. All right, so keep in mind, for an infectious agent, for a microbe to cause an infection, it has to either get on or into a cell. So this portal of entry is how an exogenous pathogen is going to enter the body. So that's A right here. Let's think of some ways. How could a microbe get inside of us? Yeah? So we could breathe respiratory droplets. That's why we're all wearing masks right now. What are some other ways that we can, I'm sorry, what are you saying?
So through food, like if it's not properly cooked or something.
Perfect. So we can have foods that contain pathogens, whether it's raw meat, or maybe some food that has some soil contamination, maybe some dirty water, or maybe even fecal-oral transmission. Maybe somebody doesn't wash their hands all the way. Or maybe they did wash their hands, but then they touched the faucet, and they get feces on their hands after they wash their hands, right? All of these are ways that we can get things in us that don't belong inside of our mouths, or our respiratory systems, whatever the case may be. So inhalation, fecal-oral, sexual transmission, whether genital-genital or oral-genital, through the skin, so again, thinking tetanus. And so all of these are ways to get in, and then organisms can cause an infection if they have what's called an infectious dose. So this is going to be the minimum number of organisms needed to cause a disease. So maybe you inhale, I don't know, 2 viral particles, and your immune system can handle that, but if you inhale 4 million viral particles, that's a really strong dose, and the infection can take place. So just think, more infectious agents is harder to deal with. Do bacteria use flagella to attach to cells? No, they don't. What do they use them for? For motility, right. So bacteria are not going to use their flagella to attach, but they can use their pili. Singular is pilus. So they can swim, swim, swim until they find a receptor, and then they can attach to it. They need to attach before they can enter into a cell. So they're going to attach using their pili. So they can attach to cells. They can also attach to other microbes and form what's called a biofilm. We've talked about biofilms a handful of times. So the biofilm is, as the word suggests, is a film of biological agents, but it also contains polysaccharides, so sugars and proteins on the outside. So this is going to help coat the bacteria with kind of like a cloak, if you want to think of it that way, to help mask them from the immune system. Not all pathogens, not all microbes can form biofilms, but those that do, that would be a virulence factor or a virulence strategy to form a biofilm. All right, so this is a really cool image. So these are some white blood cells called macrophages. A macrophage, so macro meaning big, phage referring to eat, so phagocytosis is the process by which a white blood cell will engulf a bacterial cell, or maybe a viral particle, a microbe. So phagocytosis, to eat another cell. So this is how our immune system is going to deal with exogenous pathogens. It's going to grab them, put them inside of its immune cells, and then destroy them. Well, that sounds great, but certain pathogens have co-evolved defensive strategies against this, to where they can survive within the cells that are supposed to kill them. Where are we? Come on. So cells can invade one another, or they can be consumed through phagocytosis. The difference here is that if you invade a cell, you're basically, how do I want to put this? Kind of like breaking and entering. If you were to just like do a home invasion kind of thing, for a weird analogy, you're just entering in without consent. Whereas phagocytosis is going to be like somebody caught you and then you snuck in and hid somewhere. All right, there are a couple cool enzymes that the book mentions. What type of a macromolecule is an enzyme? It is a protein. Very good. All right, so one enzyme that some staphylococci produce is called coagulase. So this is going to cause a blood clot to form. So if we have pathogens moving in blood, they secrete coagulase, we can create kind of like a blood clot with pathogens inside of it to help protect them from the immune system. This is kind of like a capsule, but it's using our machinery to form a capsule. So then, let's say, go through your course of events, and when the bacteria are ready to go back into the bloodstream, they secrete another enzyme that degrades the blood clot. That enzyme is called streptokinase. If we're talking about streptococci, if we're talking about staphylococci, it would be staphylokinase. It's going to degrade fibrin and break down a blood clot. So form a blood clot, get protection from the immune system, break down that blood clot, and go crazy, spread through the body. So for everything that we do to deal with infections, bacteria are going to come up with some sort of a countermeasure just by chance. Keep in mind that they are going to replicate a whole lot faster than us. They are really, really tiny. So their generation time allows them to compete with our defenses at a faster rate. It takes us 15, 20 years to reproduce. It takes them 30 minutes. All right, some other enzymes. Bear with me here. Hyaluronidase. Never had to pronounce that one before. So this is going to break down an acid called hyaluronidase. I can't even pronounce this. the H word with acid after it. That is located between cells. I believe it's epithelial cells. So they secrete this enzyme. It's going to break down that acid and allow them to split between cells. Leukocytins are going to disintegrate or destroy certain white blood cells. So leukocyte refers to white blood cell. We talked about macrophages. What do macrophages do? T word. Phagocytosis, yes, that's what I want you to know. So if you destroy a macrophage, then you can't be eaten, basically. So a neutrophil is going to do the same thing, phagocytosis. They can also do some other strategies that we will talk about tomorrow. Hemolysins. So hemolysis is the degradation of a red blood cell. Hemo referring to hemoglobin, lysis, rupture. The book doesn't describe why organisms would do this, but red blood cells contain iron in hemoglobin, and iron is a limiting nutrient. So organisms will rupture a red blood cell to get iron. All right, we got a slide with some more definitions here. So some pathogens can produce toxins. Think like cholera toxin, for example. So if they can produce toxins, we would say that they are toxinogenic. If those toxins end up in the blood, we have toxemia. What do we call a transient bacterial blood infection? Bacteremia. There we go. So bacteremia could contribute to toxemia if those bacteria in the blood release toxins. So if those bacteria release toxins, so that means that they're going to secrete them outside of their cells, we will call that an exotoxin. Some bacteria have pores in their cell membranes, or in their cell walls rather, that allow them to export proteins out. And some of these can kill cells, so that would be cytotoxic. Some can act on nervous tissue. We would call that a neurotoxin. And if a toxin acts on the gastrointestinal tract, we will call it an enterotoxin. And putting it all together, we've talked about gut bacteria referred to as enteric bacteria. So you see E-N-T-E-R, think gut, intestine. All right, so all these are toxins released by bacteria. Whenever our body encounters them, we are going to make what's called an antitoxin, and that will neutralize the toxicity of a toxin. Your book calls these immunoglobulins, so I'm assuming that these are some sort of an antibody. So that means that an antitoxin should be produced by a B cell, which we'll get into on Wednesday. All right, so let's say there's a toxin in our body, and there's some sort of a neutralizing event, but it still elicits an immune response. We would call that a toxoid. I can't really think of examples off the top of my head outside of what the book mentions. They mention that formaldehyde is a chemical agent that can neutralize toxicity, but still elicit an immune response, but that is the only example that they give there. Endotoxins, these are going to come from your gut microbiome. Remember we talked about LPS? What type of bacteria has LPS? Gram-negative, only gram-negatives. So, gram-negative bacteria contain LPS. This is a complex. It contains a lipid portion and endotoxin. So, the book has somewhat contradictory statements here, where they say that endotoxin alone doesn't cause an immune response, that it's the lipid component of LPS. But then the very next sentence, they go on to say that if you have endotoxin in your blood, that you can end up with a fever. So I tried to look into what the receptor for endotoxin is, and I couldn't find it in the time before class. So I'll look into that today and give you a recap tomorrow about that, endotoxicity. But I do want you to know that LPS is we can refer to that as an endotoxin. All right, so in order for an illness to spread, it's got to leave the host. So we need to make sure that we are not touching open wounds, touching urine, touching feces, right? All these things that we know, especially now during this pandemic, coughing and sneezing. In more tropical areas, things like vector-borne diseases are really prominent. So arthropods, mosquitoes can transmit disease. All right. So we have reservoirs of disease. A reservoir is going to be where does a pathogen stay within the biological world. Zoonosis is the transmission from non-human to human. So we believe that COVID-19 was transmitted from bat in an open air market to humans. So that is an example of a zoonotic disease. All right, let's spend a second on this. We've talked about horizontal and vertical transmission of DNA. Now let's talk about vertical and horizontal transmission of diseases. So what is required for bacteria to vertically transmit their DNA? What process causes that to happen? So bacteria, what's up? Vertical and horizontal. So horizontal transmission, what happens of DNA? Generally. So horizontal is going to be within a generation. So one bacteria gives DNA to another one. So that's horizontal. Now let's talk vertical. That's from one generation to another. So what's going to happen? Yeah. So for bacteria, binary fission. For DNA to vertically transmit. Okay, now we'll talk about diseases. All right, we're going to start over here with direct contact. So we can have horizontal transmission. This would be like you and I standing in here without a mask on. We could horizontally transmit respiratory pathogens to one another. With your partners, through sexual intercourse, you can end up having horizontal transmission, breathing in respiratory droplets. The whole point is that you're not going from mother to offspring. That is vertical transmission. So let's say that a woman has some sort of a vaginal infection, the baby is born, the baby can receive some bad microbiota as well. So an example would be something like syphilis, gonorrhea, chlamydia. So that's direct contact, horizontal, vertical. We can have indirect contact, things like vectors. So we can have a biological vector, something like a tick carrying Lyme disease, or a mosquito carrying malaria. Fomites, these are going to be non-biological surfaces. So really important ones are computer keyboards, ink pens, doorknobs, the things that we touch and maybe don't think about sanitizing. I saw a study once that said the most contaminated object in a hospital was the keyboard, because everybody's coming in and out and going to the keyboard. So especially during a pandemic, you all might want to take these little, alcohol swipes and clean your keyboards several times a day, especially like in a hospital, if you're not wearing gloves. All right, so a dead end host can't pass on the disease. Whereas a carrier might be someone who's asymptomatic. So again, context of COVID, we've heard about asymptomatic carriers. Those are people who don't display the symptoms of malaise, headache, runny nose, but they can transmit the disease from one person to another. So that's a carrier. So something just came in my head. I want you all to be aware that we can also have some non-infectious diseases that people can be carriers of. Are you all familiar with sickle cell anemia? Yeah? So if you are a heterozygote at the sickle cell allele, you are a carrier for the sickle cell gene, but that is a non-infectious disease. So when you see carrier outside of the context of an infection, they could have different meanings. And another reservoir could be soil or water. So wash off your food before you eat it, and don't drink lake water. A disease that's contagious we're going to refer to as communicable. So a communicable disease is transmissible. A non-communicable disease is something that you have to obtain. So something like a genetic disease, sickle cell anemia, is a non-communicable disease. You can't sit next to somebody with sickle cell and pick it up from them. You have to be born with that DNA. So here this says that they are directly acquired from the environment. So maybe something like exposure to carcinogens, working in a factory, right? I can't pick up your exposure to toxins all day just by sitting next to you. So when it says the environment, I also want to put a little asterisk next to that and say environment or from parents.
Hey, Nick,
Yes.
I have a question for you.
Yeah, let's hear it.
You're saying you cannot pick up diseases non-communicable from the environment. Let me ask you about like mesothelioma.
Mm-hmm.
Like I know a friend of the family, her dad had it and passed away, and several years later her mother came up with the same strain of that. Is that from them being in close contact and it coming home on his clothing? And
So with mesothelioma, as far as I'm aware, that comes from environmental exposure. So maybe
Right.
somebody was living in the same community. Like I know when I go through Rubber Town in Louisville, I get some sort of a respiratory reaction where it's hard to breathe. Maybe somebody wasn't a smoker, but they lived their life in the same house as a smoker, right? So this is an environmental risk that we can pick up. But as with mesothelioma, I'm not super positive. I'm going to say they probably had a similar environment, less so that they probably communicated the disease to one another.
Okay.
And what we refer to as communicable and non-communicable is subject to change. So as of right now, somebody's going to say heart disease is non-communicable. But I would argue that the pathogens that cause heart disease are communicable. Today when I get home. All right, so we've talked about this slide. This is details on it. Indirect, fomites, talked about vectors. So with vectors, we can have biological or mechanical. The difference is that a mechanical vector is going to be like a microbe that attaches to like the leg of an arthropod, but doesn't get inside of it. So it'll be a mechanical vector. Biological vector is where it's going to be within it, so like in a mosquito. All right, I think we have just 2 more slides. This might be the last one. Again, this is just big definitions before we start talking about the immune system tomorrow. So diseases occur in populations. And if there is a disease that's taking place locally, we can call it endemic. So contained to a certain area. If that starts to get a little bit out of hand, then we could call it an epidemic. Oh my gosh. An outbreak is a little bit more controlled than an epidemic. And the book doesn't give definitions like 10% of the population or anything like that, but it's just where things are starting to get out of control, but it's not like, let's start talking to the other cities about it, I assume. And then a pandemic is the state that we're in currently, where we have a global epidemic. The whole world is dealing with the SARS-CoV-2 infection. So we are in a state of pandemic currently. I hope that we never have to say that again. All right, hospital or healthcare associated infections. You don't need to worry about these with immunocompromised patients. So again, people taking steroids, pregnant, old people, people with HIV, AIDS. And so if somebody gets an infection in a healthcare setting, we refer to that as nosocomial. And so this figure right here just shows how things like airflow can cause infections to move from one location to another. And that takes us back to our control measures, talking about the HEPA filters, the high efficiency particulate air filters, to keep pathogens in one place, not spreading throughout a whole hospital. All right, and then I believe this is the last slide. So with diseases that arise, so newly in a population, we would call that an emergent infection or infectious disease. And when something shows increased resistance or comes back, we call it a reemerging or a resurgent disease. So let's say that we get control of the coronavirus pandemic, and then it comes back 2 years from now. We would call that a resurgent disease or a reemerging disease. And here's some examples if you want to take a look at it in your book. And so these can be caused by changes in land use. So things like standing water in rice fields, this is going to bring mosquitoes. Mosquitoes love standing water. Things like cemeteries have a lot of little pots for flowers. Mosquito breeding ground all day. Changes in population health, as people move, international trade. So you could have a mouse on a cargo thing, an insect on a cargo plane, and it can transmit from country to country. Okay, so as for what we are doing this week, so we're going to do 15, 16, 17 this week, take quiz 3 on Thursday, and let's get those papers done. So considering that a lot of people did not do well, reach out to me if you want help. I've had maybe 5, 6 people. I've spoken to them for an hour or so each. I'd love to help you figure out what's going on, but you got to reach out if you need help. So that's what I have for you. And so I guess at this point, class is officially over. If you want to talk about the exam, I'm going to go ahead and pull up the exam and go through it. So if you want to stick around, it'll take 15, 20 minutes. People online, I guess, you know, I'll keep an eye and make sure somebody's in so I'm not just talking to a computer. So I'm going to take a quick restroom break and then come back and we'll go over the exam, okay? So I'm going to stop sharing. All right, y'all. So hang tight. I will be right back. So what do y'all think? You want to stick around or are you going to head home? Makes no difference to me. I'm going to be here anyway. Cool. Totally understand. It's cold. All right. I'm going to go to the restroom. I'll see you tomorrow, hopefully. Reach out if you need me, okay? Good job, y'all.
So it was like, you want to do that for whatever you do. Say if I get like a 78, but then like a 85. Which I mean, I might as well do.
That was terrible. You weren't even close.
It's kind of weighted.
Wow. See? That's like, did I make it?
All right. All right, y'all. So let's see. We have about 10 people in here. So does anybody have any questions generally before we just kind of go over this? All right. So just feel free to stop me at any point. So let's go. I need to share. Exam 1. And share. Okay. So let's go through this. So y'all can see that the spread here is pretty low. Most people did not receive a C or above. Those of you did a great job. So let's talk about these. So what I'm going to go through and do is look at the questions that most people got wrong. So as I'm going through this, if you see a question that you want me to talk about that I don't talk about, then just raise it up and we'll stop. Okay. Okay, so right here I thought about putting bacteria, but I felt like that was a little bit, too easy. So I'm glad that most of us got that viruses are not alive. I wish that was a little bit closer to 100, but I'll take 85. Okay, so half of us got this one wrong. So what would you not find in a prokaryotic cell? You would not find mitochondria. Those are only going to be in eukaryotes. So the same thing would be true of a chloroplast. But all organisms contain a genome. All organisms contain cytoplasm and a cell membrane. All right, archaea, I think that this might have been from chapter 1. So no archaea are going to be pathogens. Not just for humans, we don't know of any archaea that are pathogenic at all. All right, so most people got this. This was a basic chemistry question. So atomic number is the number of protons. Mass number is the nuclear mass. So that's going to be the number of protons plus the number of neutrons. So to find out that, you would subtract 11 from 23 and you get 12. All right, when you're taking my exams, if you don't know the answer, then I will give you some sort of a potential derailing option. So I know that a lot of people will just choose all 3 if I give you that option, especially if I give you a little added little bit here, like the universal solvent. So that's me intentionally trying to derail you if you don't know the answer, and it seems like it worked half the time. So when you're taking my exams, don't look for these trends that you might follow in other people's exams, where the all 3 is always a correct answer. Sometimes it'll be correct, sometimes it won't be correct. All right, identify the polymer comprised of nucleotide monomers. This is going to be RNA. DNA is also a correct answer here. The monomers that make up a protein are going to be amino acids. Same with a polypeptide, and then with a polysaccharide, it would be some sort of a monosaccharide for the monomer. Carbon is not, pardon me, a large organic compound, but large organic compounds will contain carbon. Carbon is an element found on the periodic table. Okay, so what do all cells contain? If I said all eukaryotic cells, then mitochondria would be correct, but prokaryotes do not have mitochondria, so that's not correct. This you got credit for because you couldn't see the image. Really glad with the response rate on this one. We talked about this again today, keeping in mind that pili are used for attachment. So you all could not see this image, but this is a chain of rod-shaped organisms. So this is a streptobacillus. I'm the only one in here, so I'm going to take my mask off. All right, so the period during binary fission with the fission ring apparatus, this is going to be right before the division. So this is during D. I'm really glad that no one chose A because A doesn't exist.
I actually,
All right.
yeah, I'm gonna be the one, to be honest. I was trying
Yeah.
to choose D, and somehow I had chosen A. When I look back at that, I
I'm
said,
sorry, what do you
"Oh
say?
crap!" I said when I look back at this question, because I knew it was D, and somehow it chose A. I don't know if my hand slipped or whatever. I don't know, but
Are you
I
talking
was
about on
able-
this question right here?
Yes, the identify period of binary fission that involves
Yeah.
a fission ring. Yeah, that was me. It was, yeah, I chose A. I didn't mean to
No,
though,
nobody
but
chose
well, it says
No,
I entered
Latoya.
A,
Let me see where you're
and
at.
I was trying to choose D. I thought I had picked D, but it says that I had answered A. And I knew that you
Oh,
said
that's
that
right.
there is no period, there isn't a period. So, yeah.
Okay. So you took the exam through Teams, so yours is in a different thing here, so that's why it's showing up as 0. But I'm glad that
Okay.
now you know. That's the whole point of this is
Yeah.
to learn.
Yeah. Well,
All right.
I knew it, but I don't know how I don't know how. We had gotten a new computer from the library, so I can take it from the regular app now, or the regular
Okay.
lockdown browser.
Sounds good. Okay. This one. This was one of the trickiest questions on the exam. But I felt fair giving it to you because we had talked about it during the review. So the way that I wanted you to work this out is that if there are 2 nearby cells, they divide every 20 minutes. How many times do you have to multiply 2 by 2 to get to 128? So 2, you'd multiply it by 2 6 times to get 128. So 2 cells divide and they turn into 4 cells. Does that make sense? So each cell is going to divide. So 2 turns to 4. That's 1 replication cycle, 20 minutes. 4 is going to turn to 8. 8 turns to 16. 16 to 32. So we get a doubling every time. So you need 6 doublings to get to 128. 6 times 20 is 120 minutes. That's how you would have answered this question. Again, kind of tough. I understand. Lowest nutrient concentration, decline, right? That could also be called the death phase. An indirect counting method. Okay, so 8 people chose the streak plate method. So streak plate method is not used for counting at all. This is just to isolate a pure culture. All right, incorrect enzyme characteristics. Enzymes have a substrate. Enzymes are going to act on a substrate. They do not have a substrate. Okay, in this question, so where would you find newly synthesized ATP from ATP synthase? if you put mitochondria, then I gave you credit for this. Because what I should have done is put in a prokaryote, but since I didn't, eukaryotes will have their newly synthesized ATP in the mitochondria. So I accepted either cytoplasm or mitochondria here. All right, this is an image that you all couldn't see. This was with the Kirby-Bauer method, so these should be antibiotics. And which of the antibiotics is the most effective? We have the largest zone of inhibition here at A, so A should have been the correct, but everybody got credit for this one since you couldn't see the image. 70% ethanol is the most effective, glad to see that. Okay, this was also a relatively difficult question. So we want a really large chemotherapeutic index. That means that the therapeutic dose is a whole lot smaller than the toxic dose. Hold on. Yes, because the toxic dose is going to be the dose that is going to cause harm in a patient. The therapeutic dose is the dose that will treat a symptom. So you don't want to diagnose, or you don't want to administer a drug that could cause more problems, is basically the whole point of this. You want to design a drug that's very effective with minimal side effects. Definitions. Identify the impractical reason to design an antiviral drug. You would never want to promote viral replication inside of you. That would not be a reason to create a drug. Moreover, that is not antiviral if you promote replication. This was just straight off a figure. We talked about this today a little bit. So nobody could see this, so everybody got credit. But since we have one monophyletic group and another piece of another monophyletic group, we would call this non-monophyletic. If it did not contain this little branch and it stopped here and only included this top portion, then it would be monophyletic. But since it contains this little bit down here, it's non-monophyletic. Again, everybody got credit for this, though. This is the case where the all option was correct. Definitions, definitions. Again, this is one where all three was the correct answer. Just more definitions. And then we had talked about this 2 days before the exam. So if a virus is able to bud out of a cell without killing it, that is going to allow for more viral particles to be released because you're not undergoing lysis and killing the host cell. So more replication can take place, and if you have more replication, then you can do more exogenous spreading to new hosts. So that was the exam. There was a whole lot of questions in there. Everybody got at least 4 out of 4 correct. I was really happy with a few of you. You did really well. For those of you who didn't do as well as you thought, we're going to keep working hard. And I'm thinking of potentially doing like a, oh my gosh. Oh wait, I am sharing my screen, sorry. Stop sharing, there we go. So what I'm thinking about doing is a cumulative makeup exam where if you want, you'll have the option to take an exam after your second practical that will cover the entire course. And then you'll get to drop your lowest exam score. That's kind of where my head is right now, because I don't want to curve. Because if I curve, then that takes away the high grades from those who studied hard, and it also gives people who scored very poorly a higher grade without earning it. So I'm not going to curve. I don't want to do that. What I'd rather do is give you a chance to redeem yourself and to work hard and learn the material, because that's really what this is about, is learning. So how do you all feel about that as an option? Yeah?
Yeah. If
Yeah,
you were to give
Okay.
that, I would take it.
Okay, so we'll do, my idea is after exam 2, so I might need to do some reorganizing of the syllabus to accommodate this. So a makeup exam, optional, and then we're going to do Tuesday, Wednesday, and Thursday brief review sessions before the start of class. And so that's the changes that we'll make this week, and we'll see how it goes. And so if you all have anything that you want me to do or that you don't want me to do, let me know during class or shoot me a little private email, whatever it is that you think might be helpful. I'm more than willing to accept your suggestions. If you want to go over the quiz from 2 weeks ago, whatever it is, just let me know. If you want to talk one-on-one, let me know. So that's what I've got for you all. Does anybody have questions or feedback, anything right now? Questions about the paper, anything?
I do actually have a question. There was one last question that you went over on the quiz. I didn't catch which one it was, though. I think it was the one.
You're talking about on the exam that we just talked about?
I mean, yeah, on the exam, sorry. I think it was the one, identify the true statement regarding viral spike protein.
Okay, yeah.
I don't think that is that the question that you went over last? The very last one?
No, I don't think that I had discussed this one. All right, so the true statement regarding viral spike proteins. Okay, so they can be present on non-enveloped or on envelope viruses, so it's not either of these. They do not contain genetic material, but they do help viruses attach to host cells.
Okay.
So the viral spike protein, you can think of, it's similar to a pilus. Or, you remember, so bacteria have they have the pilus, which is an extension from the cell wall. And on the end of the pilus, they have the adhesive domain called an adhesin, okay? So you can think of an adhesin from a bacterium as analogous to a viral spike protein. So it's going to be on the outside of the cell, and it allows it to stick and attach to a host cell.
Okay. Like the coronavirus thing. Because it has
Yes.
those, like the red things sticking out on the outside of the
Yep.
cell wall.
Yep.
Okay.
That's going to be a spike protein.
Okay. I will email you a little bit later regarding the whole thing for the session for me. It's really hard for me to focus while I have 2 small children. So I will email you
I can only imagine.
later. Yeah, it's difficult. It is.
No,
So
I get it. So
I'll email you about
you
that.
just shoot me an email when you have something together, and we'll be in communication over the next couple of days, okay?
Okay, thank you.
You're welcome.
I have a
All
question.
right, anybody have anything else? I'm sorry, what's that, Sabrina?
I have a question about the paper.
Yeah.
So I found the study that I want to put into my paper, but I'm having a hard time understanding it. So can I bring it with me tomorrow and have you help me? Because I'll be in
Yeah,
class.
sure. I'll be in maybe like 20 minutes early. I'll be in at like 9:15, 9:30 tomorrow. Are you going to be in early?
Oh yeah, I'll probably be there at like 8.
All right, cool. I'll see you before class starts tomorrow then.
Thanks.
If you want to email it to me tonight, you can do that too.
Oh, okay. Cool.
All right, y'all. I'm going to go ahead and end this recording. If you need me, go ahead and reach out. And since Canvas is full, I'm going to post a link to this on my YouTube page and then post that link on Canvas for you. And then keep in mind that the slides for today are in an email attachment. So I'm going to go ahead and end this, and I will either talk to you all today through email or see you tomorrow in class. Thank you all.
Thank you.
Bye.
Bye.